Hello, this is Dr. Comparo, and today we're going to talk about thyroid hormone synthesis. We're going to talk about how you create T3 and T4 in the body, in the thyroid. So first of all, let's locate the thyroid. It is located in your neck, and it's a very small gland that looks like a butterfly. A very weird butterfly. And that is located in front of your trachea. So you're anterior to there. And this is the gland that controls most of the metabolism that occurs in your body. So now I'm going to show you the functional unit of this gland. It's called a thyroid follicle. So it kind of looks like a jelly donut. It's got a layer of cells on the outside. These are all different cells. And these cells, they are called thyroid follicular cells. And of course, they have nuclei and all that. And then in the middle, what I'm calling the jelly, this is called colloid. This is a protein-rich, kind of gelatinous glue. And so these little things here, these are follicular cells. And then right here, we have colloid. The whole thing is called a thyroid follicle. And so now we're going to take a look at a chunk right here at the cell wall. We're going to look at one of the walls of the jelly donut, if you will. And I'm going to draw that on a different screen for you. So we're going to have the cell wall here. This is the lumen where we have the jelly filling. These are the follicular cells. And these cells, they produce a protein and they spit it out into the lumen. This protein is called thyroglobulin. And it's secreted by the follicular cells via exocytosis into the lumen of the follicle. This is a very long protein. It's a peptide chain, and it has a lot of these little rings called tyrosine rings. So a bunch of these are coming off of here. I apologize, my drawing's not quite the best today, but I think you get the concept. All right, so we have this kind of sitting in the pool. Now, iodine in the body is called iodide because it's an ion. That is going to be taken into the cell via a receptor called a NIS transporter. NIS transporter, and it's called NIS because it is a sodium iodide symporter. So it's just an abbreviation. This is going to bring the iodide into the actual cell, the follicular cell. Then, after that's in there, it's going to go through another receptor to go into the lumen, basically join the pool party. And this receptor that brings it into the lumen is called a pendrin receptor. Once the iodide is in the lumen, then it's going to be converted to atomic iodine. So now it's called iodine instead of iodide. And this transformation is facilitated by thyroperoxidase. Whenever you see ACE, that means that something is being metabolized. This is an enzyme, so it's being broken down. Thyroperoxidase peroxidase is going to actually oxidize the iodide ion to form iodine atoms. So these atoms, they like to bond. And they can bond in two different ways. They go over to the tyrosine, and they can bond either single or double. Okay? And so if it binds just singly, we call that MIT, which stands for monoiodotyrosine. And of course, double is DIT, which is diiodotyrosine, right? And the tyrosine molecules, they can join up with each other. They kind of fold in on themselves. And you can do it in a combination of either one MIT with one DIT, like we have right now. And so if you count the iodines, we've got 3. So that's what we call T3 or triiodothyramine. That is the principal thyroid hormone. That's the one that does most of the action of the thyroid. Now, let's say we had another iodide here, and you had this combination, then you have T4. And T4 is tetraiodothyramine, or we just simply call it thyroxine. So that's how you get T3 and T4. So T3 and T4, they're going to be taken into the follicular cells. This is endocytosis, and it's going to be still attached to the thyroglobulin. So it's still on the tyrosine rings, but now they've kind of bonded together. The whole thing is going to get brought up into here, into the cells. And then it's going to be broken down by a bunch of different proteases. It's going to undergo proteolysis. And pretty much it's going to get broken down just into T3 and T4, which is then going to be excreted from the cells, secreted into the bloodstream. So that would be a blood vessel up here. So of course, as you probably know, T3 is the active form of thyroid hormone, and T4 is the inactive, but it acts as a storage because pretty much all you have to do is lop off one of the iodine atoms, and then T4 will become T3. And the enzyme that facilitates this is deiodinase. So that is how you get T3 and T4 synthesized and how you get it released into your body. If you have any questions, please leave a comment. I would love to elaborate, and thank you very much for listening.